I want to mention that we have set the date now for the next science conference for AnaEE-ERIC. There is more information on the website, and I encourage everyone to subscribe to our newsletter, because that's the best way to get news and updates about this. But now let's begin this short introduction to the first direct call for AnaEE-ERIC research facilities, and to AnaEE-ERIC in general also. I'm the communications officer for AnaEE-ERIC. We have 4 centers, and me, I'm based in Paris and within the central hub. And so I will just tell you what we are going to talk about today. It will be a short introduction to what is AnaEE-ERIC, what do we offer, what are we all about. Then Dario DeNart, my colleague, will take over and explain what data and modeling can offer researchers in the network of AnaEE-ERIC. Then we will present some other facilities. It's enclosed and aquatic facilities, and we also have open air, but we are not presenting them today, but maybe in a later event. And then I will show you where you can find resources, how you prepare and how you apply, and questions and answers at the end of the webinar. So you can say that AnaEE-ERIC is kind of a European collaborative organization and research network. We have about 25 research institutions and universities spread all over Europe with their unique facilities offered within the network. Our specialty, there are research infrastructures within many different categories, from medicine to infrastructure to IT to space. And our specialty is then how ecosystems respond to various pressures, from climate change to pollution, and the best ways to mitigate the risks. And so we offer services, and we offer resources to policymakers, or if you work with nature-based solutions, green technologies, if you come from the industry, we can also offer you resources. Or if you are maybe working for an institution or a university, there is a lot of resources through AnaEE-ERIC. You can just scout our website and contact us if you're interested in finding out more. So now I will briefly explain the facilities to you. So our facilities have advanced equipment that can simulate future environmental conditions, such as soil composition, drought, CO2 levels, and we can observe how plants, animals, and entire ecosystems react in all European climatic zones. And we also have some tropical facilities in tropical climate in the South America. And so we have 4 different types of facilities. They are grouped into open air, which is basically like a field where you can grow crops. Enclosed, like this one in the picture, which is very enclosed. And then we have modeling and analytical facilities as well. And here you can see gives a good overview of the spread of our facilities. So it goes from the south of Italy to subarctic climate in the north of Finland. And it's managed as well as unmanaged land, agro-ecosystems, peatlands, forests, etc. And so that's really a short introduction to AnaEE-ERIC. And I will tell you later how to apply funding and where you can find more resources about the call. But now I will give the word to Dario DeNart, who is the IT manager at CREA in Italy, and he will explain to you what data and modeling in AnaEE-ERIC can offer you.
Talk very briefly about what we do in terms of managing the data and the models in AnaEE-ERIC. And spoiler, the end game here is to have a good EOSC integration. That is something. EOSC, as you might know, is the European Open Science Cloud. That is what the European Commission is asking more and more to research institutions. But it is not easy, because it takes an IT infrastructure, it takes dedicated personnel that not all institutions have on site. There is this very big push towards federating with EOSC, pushing data and services into EOSC, and it's becoming a requirement in the Horizon project. So it's becoming something that all universities and research institutions will have to do somehow. And on top of that, the EU is already funding a lot of initiatives. So it is, let's say, legitimate expectation from them to have universities and other research institutions to comply with these demands. But the sad truth is that nowadays, at least in our experience, most institutions are not really ready. And there is where AnaEE-ERIC can come into play, because we are very well positioned in this environment. And actually, in the ENVI cluster, we are one of the leading infrastructures when it comes to IT development. And because we have a dedicated competency center, that is the data modeling center, that is located in Italy, that has full-time and dedicated IT personnel. It can aggregate, it has services, online services that can help your institution achieve compliancy and data fairness. And we already participate in EOSC-related projects. One is the ENVI Up Next project, in which I am work package leader of 4 work packages. And we have also a growing service portfolio that is supported by EU funding and member state funding. So we have dedicated funding for that. So what do we do then? We focus really on translating models and data sets into services. Here I'm talking mostly now about models because it's easier to understand how things work. So usually when you as a researcher code a model, you do that in your virtual research environment that might be either in the cloud in a solution like Collaboratory or something like that, or it can be locally like RStudio, Jupyter Notebooks. The problem is that most of the times we see that there's lots of hard-coded stuff and there's very poor reuse potential. What does that mean? It means that once you're done with your experiments, with your paper, it's very hard for the rest of the scientific community to benefit from that. So the vision of AnaEE, that by the way is also the vision of EOSC, is to turn that research code into web services that expose APIs that are application program interfaces. It means that your code becomes something like a function that lives in the cloud. People can send them queries and return and have responses in return, like you can ask for a specific fragment of data, you can ask a model to run on a specific subset of data, and then you get back either the raw data or the simulated data. So how does that happen? So we can support you as data modeling center into this transition towards from research code to research services, because we have the infrastructure to host these services. So once you packaged your code, you can run it on our servers because we have a Kubernetes cluster. We are proficient with virtualization technologies. We can give you some degree of assistance if you want to create your own on-premise cluster. And on top of that, we also provide an API portal for them. What is that? It's a website where all the APIs developed by the AnaEE community are listed. So there's already something in there. We can put your services in there. That it acts like a catalog. There's the metadata. It allows people to find your services. But most importantly, it handles for you public IPs, login of users, user authentication, user authorization, logging, telemetry, throttling, and a lot of other services that otherwise you would have to code on your own. And so you'll find them for free there, basically. And so you don't have, if you load your services on our infrastructure, we can publish them there, and you don't have to care for scalability and a lot of other technical issues that researchers are totally unaware of. So what's left for you? What you have to do is to make refactoring of your code, which is actually very simple because concretely it's mostly annotations. So if you can see here, this line with the @ symbol, basically it's adding a lot of this in your code. And we can help you because we know the frameworks. So you can ask for training about that. You can do the same thing for not just for data, but also not just for models, but also for data sets and even for visualization services. Everything can be turned into a REST service. And the benefits are that there's a better interoperability and reusability. So you know of fair data. There's the first two part, findable and accessible, usually are easy to achieve. Interoperable and reusable are very hard, and this is the industry standard to get these two properties. So an example, a very simple example, is that you can, if you have a database with your records, you can write an API, we can write some code to expose that. So you don't give users full control over the database, but you can select what they can access and what they cannot. And more importantly, you prevent them from making disruptive operations. Some projects are already doing this. For instance, there's the BGuard, it's an Horizon project that is exposing like this its IoT data and has it already in the AnaEE portal, as you can see here on this on the picture. And also visualization applications can be exposed, and the transition from notebooks to web application is made easy by tools such as Shiny and Dash, that are tools we are quite familiar with, and we can offer you support. So the question is, do you want to be part of this transition? Because if yes, AnaEE can help you. You can, I mean, if you are very skilled with that, you can become an analytical platform. If you're not yet skilled, you can get training, you can ask for supports, and the end game here is really to create a constellation of AnaEE services that will be fed into the European Open Data Science Cloud. And one final word, this is not just, pursuing a dream of a better open cloud, but it's a very concrete thing that goes into funding, because European Union is expecting people to provide resources for the European Open Science Cloud. That's it.
Okay, excellent. Thank you very much, Dario. And now I will leave over to the next point, which is about Ecotrons, simulating the future. And the Ecotron is just one type of many different enclosed facilities that we offer in AnaEE-ERIC. And it will be presented by Alexandru Milcu from AnaEE-France. He is the director of the Montpellier CNRS Ecotron. He is an ecosystem ecologist whose research interest focuses on the consequences of global changes such as biodiversity loss and climate change for the functioning of terrestrial ecosystems.
Yes, today I have the pleasure to present some of the experimental facilities. We've been hearing about the modeling capabilities of the AnaEE Europe. The experimental side of AnaEE Europe is a very important side as well. And here we have facilities that offer experimental systems in natura, like in the field, but also control environment ones, like the Ecotron facilities that I'm going to talk about. And there are several of them in AnaEE-ERIC, and I will start by explaining a little bit the principles of the Ecotron facilities, how they are different from typical phytotrons on greenhouse gases, and what are the experimental trade-offs that I think the community is perhaps overlooking a little bit. And yeah, eventually, what are the typical capabilities and why would you choose an Ecotron? So what is an Ecotron? Because I mentioned that it's very different from a greenhouse. It looks like a series of greenhouses. And very often the climatic simulation aspect is put forward. But this is one side of what an Ecotron is. So yes, Ecotrons can simulate various climatic conditions much better than a typical greenhouse. What is different is the fact that these facilities have been designed with the capacity to measure automatically and continuously ecosystem processes. This is what makes them an Ecotron, these both capabilities. So the environmental control and then the automatic measurements of ecosystem processes. And what kind of ecosystem processes? This will depend a little bit on the Ecotron. Each one has its own focus and specificity. But typically, we're looking at ecosystem carbon fluxes, so how much carbon is fixed by the plants, what's the overall ecosystem respiration, as well as emissions of other greenhouse gases like nitrous oxide and methane, as well as water fluxes and isotopic label and tracing. And of course, in any experiment, you can, and typically we have the additional experimental treatments on top of it. So sometimes we just simulate climate in a realistic way and have other experiments, other manipulations on top. Sometimes the environmental control, the climate itself is a treatment, but that's very experimental dependent. What else is important to remember is the fact that an Ecotron has always replicated controlled units. If we just have two of them, we cannot separate the effect of our treatment from the effect of our experimental unit. And this is a very important distinction. And also, it is positioning itself as an experimental facility somewhere in between the uncontrolled conditions of field studies and the oversimplified conditions of laboratory experiments, typical microcosm pot-size experiments. And there are many trade-offs when we choose the kind of experimental approach we want to tackle our hypothesis with. And I know this slide it's complicated, but on the upper part here, we see a range of experimental approaches from very simple lab, small scale, artificial, oversimplified microcosm or pot experiment to the sort of Ecotron-like, larger in size, more realistic ecosystems, to the uncontrolled conditions of the field sites. And what varies here a lot is the level of control. So we have much better control over what we want to explore towards the smaller scale. And but at the same time, there are other trade-offs. We'll come to them. Another advantage of going towards this small scale size is that we can have a lot of replication, is cheap, is affordable, is easy to do. And this is great because we can have a lot of replication statistical power. And also, because we can control very well our parameters here, we can isolate the mechanism we're interested in and change that particular variable that we're interested in. So we often say that these experiments have high internal validity, so this capacity to disentangle the mechanism. But there are a lot of trade-offs, and I think these are overlooked. One of them is that if it's so simplified and sort of artificial, we are losing a lot of key properties of natural systems, which we have if we go towards this end of the experimental approaches. And also, we're losing in the so-called external validity if we are on this extreme. The external validity is the capacity to generalize the findings to other systems or to field-like conditions. And this is where the experimental field sites are pretty good in terms of external validity and realism. They deal with very complex natural ecosystems, but they lose in this capacity to disentangle mechanism. We don't control a lot of things. We won't be able to necessarily indicate and understand that particular mechanism. We will measure effects that are real, and we can transfer them. And this is where the Ecotrons are somewhere in between, because we can have a little bit of good things of both worlds. We can control things, but also we have experimental systems that are rather large and complex and simulated with high realism, so that what we find here is usually transferable to field-like conditions. So in AnaE Europe, we have several Ecotron-like facilities. Like I mentioned, each of them with their own particularities. I'm not going to go into detail. You can go on the platform and explore the different kind of Ecotron facilities that are offered. But I will continue just a little bit to present what we have at Montpellier, because it will give you an idea of what these facilities can do. So the Montpellier Ecotron, we developed 3 experimental platforms: macrocosm and microcosms and mesocosms. The mesocosm and the macrocosm are really typical, like fully fledged Ecotrons. The microcosm are somewhere between a phytotron, a typical growth chamber, and an Ecotron in terms of capabilities. So if you're interested in knowing a little bit more about what the Ecotrons are doing, you can have a look at this paper, this review that we published in 2020, where we present the capabilities of all facilities that at the time fit the criteria of being an Ecotron. Like I said, just having a phytotron or a greenhouse, it's not an Ecotron. And here I emphasize the continuous ecosystem measurements that we can measure in the macrocosm and mesocosms. You can see here a wide range of automatic measurements in terms of ecosystem processes, but I'm not going to go into more detail. So just very briefly, the macrocosms, where these are the most realistic in terms of size. We are getting close to a small field plot. We're talking about 5 square meters of experimental surface per unit, and we have 12 of them, with a soil depth of 1.5 meters, roughly about 14 tons of soil in each system. We can control plus minus 10 degrees Celsius from the outside temperature. We can also impose and control a soil temperature gradient that is realistic. That's something a lot of small pot experiments cannot do. Obviously, we can do the typical control environment, humidity and temperature, elevated CO2, automatic precipitation. We also went for natural light with the dome that is using a transparent material that leaves most of UV passing through, because the UV is very important in terms of plant phenology and development. And yes, this continuous high frequency measurements of ecosystem processes. And this is how they look like. These are experiments that are quite heavy to set up and require resources to not only run, but also to set up the lysimeters, the containers with the soil, which can be done either with intact soil monoliths from the field or reconstructed soil profile. Now, the other platform, the mesocosms, it has basically the same principles as the macrocosms. I forgot to mention that these greenhouse gas fluxes are measured a little bit like a Lycor automatic gas flux chamber. They are designed in a similar way. In this particular platform, we have high replication, 1 square meter surface experimental units, 1 meter depth. We could do also pre-industrial carbon dioxide concentrations and also ozone atmospheric control. And finally, in the microcosms, these are, like I mentioned, sort of phytotron type chambers with some capabilities that are approaching Ecotron-like facilities. And they're very versatile in terms of experimental setup. You can see here some of the experimental setups that we have. We have 13 units here with LED solar simulators, capacity to use certain sea labeling, and in general, they are sort of more affordable and cheaper to run. But we run into these trade-offs in terms of generality and capacity to extrapolate to field-like conditions, which is more limited in this kind of setups. So this is my last slide. I want to, again, emphasize these trade-offs because I think very often we are tempted to go with the easier setups, which very often is a few pots in a greenhouse or a lot of tiny little experimental systems in a phytotron. But I think we need to be aware of these trade-offs because we want what we actually find to be transferable to natural systems and not just measure something that is true in that particularly oversimplified system. So I'll leave it to that, and thank you very much for your attention.
Thank you, Alexandru. That gave everyone, I think, a good idea of what sort of capacity you have in this type of facility as the Ecotron. And so now I will leave over to Thomas Davidson. We are moving over to Denmark. He's a professor in freshwater ecology at the Aarhus University. He's a freshwater ecologist and biogeochemist and a leader of the Silkeborg Lake Mesocosm experimental system. So it's an aquatic facility.
So yeah, thanks to Alex for a really great overview of the costs and benefits of Ecotrons, and I'll talk a little bit about our aquatic mesocosm, which I guess is a type of Ecotron, or has become more of one as we've added more analysis types to and more high-frequency continuous measurements of various parameters in the system. And coming from Denmark and the land of Carlsberg, I don't know if you remember the advertising campaign, but I borrowed it here to say that our system is probably the best aquatic mesocosm facility in the world. Probably is doing a lot of work there. So I'll just give you a brief introduction to it here. It lies in the center of Jutland, which is the part of Denmark attached to mainland Europe. And it's in a place called Lemming. It's our climate change and nutrient enrichment experiments. And we, in our house, we call it the Lemming experiment, which can lead to some confusion because it's got nothing to do with the Arctic animals, but it's based close to a little village called Lemming. And it looks like this. It's 24 small lakes treated. And we have the treatment was set up in 2003, and it consists of 2 nutrient levels. So on this figure here on the left, we have where it has a plus, we add nutrients, nitrogen and phosphorus every week to create a kind of eutrophic system. And then where there's no pluses, so the A's are nutrient enriched, the D's, and then the B tanks would be low nutrient levels, and they receive just groundwater input, which does have some nutrients in it, of course, because groundwaters have some nitrogen in it, particularly in Denmark. And then we have 3 different temperatures. So in the white, we have the ambient temperature. So each of these clusters of 3s are controlled together. So the white is the ambient temperature of central Denmark. And then the green, it was the A2 scenario of the IPCC assessment at the time the experiment was set up, which was early 2000s. So it's 2 to 3 degrees warmer than ambient. And then there was an A2 plus 50% treatment as well, which is 4 to 5 degrees warmer than ambient temperature. So we have nutrient enrichment crossed with 3 different temperatures. And this is what the tanks end up looking like. So at low nutrient treatments, we have clear water. These are the rather extreme ends of the gradient. We have some within treatment variation. We have 4 replicates within each of the 6 treatments. So we have clear water conditions with abundant plants in low nutrients, and we have turbid conditions at high nutrients. So this allows us to look at how climate change, or at least warming associated with climate change, interact with nutrient enrichments in fresh waters. So this image of the mesocosm on the top left shows you how the systems were set up. Sediments were homogenized from various different sources, several lakes and mixed, and placed in each tank equally in 2003. And then we have a meter of water above, and then we have like 0.3 meters of lid to the tank, and the diameters are 1.9 meters squared. So they're around 2,000 2,000 liters in the tank, and they are flow-through systems. So we have a groundwater input and an overflow. They have about a 2 months residence time. They also have a mixing paddle to stop. You can't see it on the diagram here, but here you can see it. They have a paddle which moves and mixes the tank to avoid stratification. And then the heated tanks are heated by heating elements here. And they're regulated in groups of 3 with a reference. So each warm tank uses the ambient tank as a reference and heats to 2 to 3 degrees. And that changes seasonally as the climate change, downscale climate change predictions estimated that the warming would be greater in spring and autumn, basically. And we continuously measure oxygen, pH, since the beginning of the experiment, so we can calculate net ecosystem productivity through diurnal variations in oxygen. And as a part of the AnaE project, we actually got funding to add chlorophyll and phycocyanin measurements, continuous measurements to the tanks in, I can't remember exactly where that was, when that was, but about 6 or 7 years ago. And they've been functioning extremely well, actually, since then, after some initial difficulties setting up. So it's actually the longest running aquatic climate change experiment, running since 2003 continuously. We've done some extra experiments over that time to look at multiple stresses, including a heat wave for short periods, a DOC addition, a nitrogen pulse. We tried a stratification experiment in 2001. And we also stopped the addition of nitrogen for 1 year in 2018. We were part of several EU projects, Aquacosm and Aquacosm Plus, and we delivered over 1,200 days of transnational access during those projects. So we've had a lot of guests coming to work on different aspects of the experiment. And in 2023, after 20 years of the project, we actually reversed the nutrient treatments. So the formerly low nutrient tanks have been having nutrients added since 2023, and the formerly high nutrient treatments we stopped adding nutrients. So we can study the oligotrophication processes and the eutrophication processes in real time. And the things we've added in the last couple of years have been the high frequency measurement of greenhouse gas emissions. And we've built a kind of low cost system, which we won't go into the details of here, but we've got tanks on all 24 experiments. And we used some of these low cost methane and CO2 sensors and a low cost flushing system to provide kind of diurnal variation. Here we can see 2 days of data. So we have CO2 in black and methane estimates. This is the raw output of the sensors, actually, so it's not ppm or anything. It's easy to understand, but we've calibrated them and we can convert them to that. And here we can see this is the CO2 increasing as in the night when the tanks are sources of CO2, and this is the flushing. And then in the daytime, we can see that the tanks are actually taking up CO2 over a couple of days from June, and that the methane emissions are kind of quite variable over the diurnal cycle. So we have continuous measurements, and this is what happens when you take like a 4-day period. So we have 6 estimates of flux of methane and CO2 flux over time, and this is basically to show you that we have high temporal variation in methane emissions over 4 days here. And actually the top here is the kind of the atmospheric concentration of methane, and you can also see that we have some nighttime increases in atmospheric concentration of methane in the locality as well. So the background varies, and this is actually pretty interesting to think about too. And then just to show you how this information, high frequency information, is then converted onto a yearly scale. Here we have the daily, actually the 4-hourly estimates of CO2 emission from one of the tanks over the course of a year. And we can see that and then the green line, we have the continuous chlorophyll A measurements. So we can see how the fluxes of CO2 correlate very nicely or are very strongly related with chlorophyll concentrations, and that the tank is mostly a sink of CO2 with some emission events that correspond to like quite sharp drops in chlorophyll concentration, which happen for one reason or another. I don't have time to go into that, but we can also see that at this tank that had a high PVI. So this is the percentage of macrophytes that was very high in the summer, and this also contributed to CO2 uptake in the tanks. So we can get this very nice high temporal resolution study of the patterns of greenhouse gas emissions and their drivers over time. And then what would be the opportunities for collaboration? Well, given our current experiment, we have really good opportunities to look at the effects of nutrient change at different temperatures. There's a lot of possibilities to study the benthic systems within the tanks, because like biofilms and processing of nutrients in different areas of the tank and of carbon. We have real sediment in the tank, so it's possible to do in situ studies, and we have a lot of supporting data on plant abundance and chlorophyll and blue-green algal pigments and oxygen and the greenhouse gases. So there's really a lot of possibilities for more detailed studies. And with that, I thank you for the time.
Thank you, Thomas. That was a very nice, gave a nice introduction to the capacities of this aquatic facility. So maybe these 3 presentations made you curious about applying. So how to do that then? I will show you where you can find all these resources. The call is open now, as you might know, and the resources are available through our website. So you just simply go to the first page. There is several ways to find it, but you can click open calls. And here, AnaEE-ERIC, and the call is open until, you can apply until October 3rd. Here we have some general information, the link to the ECI catalog, the application form, or you can click here for users access policy, guidelines, and the step-by-step, like the evolution of the project through AnaEE-ERIC. Here is what you have to think about before you apply and after your project is finished. And here is the AnaEE-ERIC project evolution. Then we have the users access guidelines and policy. It's a PDF, you can download it, have a look. And here is information about all you have to think about. And you apply through ECI. And the catalog of services to see what sort of facilities that are available. You can go also to the website, you click services, and you click here you can click services here and click catalog of services here, or we have it here in the menu. And here it's possible to search by scientific domains, scientific subdomains, free text keywords. So in free text, that's right, CO2. There is a bit more than 60 facilities available. And application portal is pretty straightforward. I mean, it's an online form that you fill in step by step. Okay, so how then be a successful applicant? Well, A and O, like the most important is that when you find a facility that you are interested in using for your research, you have to contact the facility manager to discuss with him or her if your project is feasible. Is there a way you can change a bit the project, or that you could use the facility more efficiently than you initially thought? So they are there to help you and improve the project scope to be the best possible. And also a question that comes often is, what about the price to use the different facilities? How much does it cost? It's the same thing. When you contact the facility manager, they will be able to provide you at least an idea of what the price tag might be to use the facility. And if you are not sure that you can find the funding to do the project, you can always reach out to us, and our team is here to help you. Maybe we can find a way to link your project to one of your research project to one of the European Horizon Europe projects that we have, and there are open calls coming and going throughout the year. There is AgroServe, AquaServe, etc. So different facilities are available through different projects. Okay, but now you should know where to find more information and resources about how to apply, and I will open for questions and answers. So here is from Janko. He has a question regarding Ecotrons to Alex. How long do experiments in the Ecotrons usually last? Is there also a possibility to install sensors monitor root growth, development, and competition?
It will depend. The answer kind of always is, it will depend on your scientific question. So, but it also depends a little bit on the platform. For the macrocosm, mesocosm, Ecotron systems, because the setup is very sort of heavy and time consuming, typically we also run these experiments for a few years. The other reason is that we don't really want in this, what we claim to be more realistic systems, to actually measure short-term transient responses. So typically macro and mesocosm experiments run 2 to 3 years in average. If you want to tackle your question using experimental hypothesis using the microcosm platform, the phytotron type facility, there typically experiments last 2 to 3 months. Again, I'm averaging. It can be longer, it can be shorter. And yes, this is a very important point. We can add and install additional sensors. Actually, every single experiment, it's custom designed with the scientist to best respond to the respective scientific question. And very often we have to install new sensors or adapt. And yes, Rhizotrons, it's something that can be installed. We currently do not have a system in place that can do that automatically. I don't know what kind of Rhizotrons you have in mind, whether with automatic camera recording. But all of these things are possible, and we typically develop them with the team of scientists that come to work with us. Thank you.
Now it's 11:47, so the time for the webinar is up. And if you are interested about more, we are here. The AnaEE-ERIC team is here to help you with any questions. We have our excellent scientific officer, for example, might be able to guide you further if you have any questions on what sort of how to form your experiment. So have a nice weekend, and thank you for participating.
Thanks.
Thank you very much.
Bye-bye.
Bye.
Thank you. Thank you very much.